Hi everyone, this is Christy Weeks, and I am your instructor this semester for the BTC 250 Principles of Genetics class, and this is our very first lecture on Chapter 1, Introduction to Genetics. Now, like every other science course you'll ever take, the very first chapter is going to be an overview of pretty much everything you're going to learn throughout the semester, and so it's just going to be a really broad stroke and just kind of go over the surface, and this chapter is no different. So we'll just get through that, and if anything seems a little like too much information all at once, don't worry, we're going to be delving into all this stuff much deeper with subsequent chapters. And I will probably say over and over again throughout this lecture that we'll talk about this in more detail later. We'll talk about this more detail in chapter XYZ. So here we go.
So I think the book gives a really good example of how we can apply what we've learned in genetics to medicine. They give the example of translational medicine, where we use the information that we have discovered from studying our genes to figure out what causes disease and ways that we can treat disease on a molecular level based on genetics. And one of the examples that the book gives is the PCSK9 gene and how it affects LDL levels. So, we know LDL cholesterol is an important factor in heart disease, and there's several drugs on the market that lower LDL. But this is kind of an alternative way to lower LDL. What happens in liver cells is LDL, the cholesterol molecule, actually binds to a receptor on the cells called the LDL receptor. And we know that there's a disease called familial hypercholesterolemia that's associated with a mutated form of the receptor, and so the LDL doesn't bind to the receptor and is not taken into the cells, and the cholesterol remains in the blood, and so you have really high cholesterol levels. But to make things more complex, you can have a perfectly fine receptor, and the receptor will bind to the LDL, and then it will pull the LDL into the cell. But there's another protein that's involved in this process called the PCSK9 cell. So once the LDL is brought into the cell, it gets broken down and recycled by the cell. And then there's this protein, PCSK9, that then can bind to the receptor and actually degrade the receptor. And mutated forms of this protein can lead to the receptors being broken down too frequently, so that there's not enough receptors on the surface of the cell to bind the LDL, and so again you'll have high cholesterol levels. And with this knowledge that we've gained from this enzyme, we've been able to develop antibodies that bind to the enzyme, bind to the protein, and prevent it from binding to the LDL receptor. And so that creates a surplus of receptors so that more LDL can be brought into the cells from the blood, lowering the blood cholesterol level. So this is just a really good example of how the information that we've gleaned from genetic studies and learning about the function of our genes, how we can use that information to treat disease. Okay, so now we're just going to do an overview of the history of genetics and talk about some of the main players associated with genetics. So we've always had the ability to domesticate animals and plants, and so the livestock that we have today and the plants, our crops, are due to effects of selective breeding that we've carried on for the last 1,000 years, and that's a form of manipulating genetics. Aristotle and Hippocrates discussed heredity traits long ago. They both knew that traits were passed. I mean, people, since we've been people, we've always at least been able to observe that our children look like us. So we've always known that traits get passed down. And Hippocrates argued that there were these humors that that's where the traits came from. And he even argued that these humors could be altered by the environment. So that was, that was pretty good that he figured that out. And Aristotle believed that there was some kind of vital heat present in male semen for producing offspring, and that's how the offspring came in the same form as the parent. So, well, that's not really true. They're at least observing genetics. So 1600 to 1850, that's what we call the dawn of modern biology. In the 1600s, a guy named William Harvey proposed the theory of epigenesis, which is the idea that you have this organism that's going to develop from a fertilized egg or a fertilized embryo, and that it develops from an embryo to an adult. Now, this is different from what the previous theory was, because they believed that literally there was this tiny little human inside of either the sperm or the egg, and that tiny little humus called the homunculus would just get bigger and bigger until a baby was born. But it was actually completely formed with arms, legs, and eyes and everything from the get-go, which is kind of crazy. So in 1830, that's when the idea of the cell theory came around, when they were able to start looking through a microscope and see cells. They recognized, Schilden and Schwann recognized that all organisms are composed of cells, which are derived from pre-existing cells. And this was counter to a previous theory of spontaneous generation, that life just kind of spontaneously generated itself. And Louis Pasteur was the one that disproved that theory. And in fact, if you take microbiology, you'll learn all about Louis Pasteur and how his very ingenious experiment that he did to show that. Now, everybody knows who Charles Darwin was. So he traveled all over on his boat, the HMS Beagle, spent a lot of time in the Galapagos Islands, and through observation formulated his theory of evolution. And so then he published a book, The Origin of Species, in 1859, and he believed that all of the species that are present on the planet arose by something called descent with modification, and that's based on the theory of natural selection. So anytime there was some kind of change in the animal that gave it some kind of advantage, an ability to adapt to its environment, then those would be the individuals that could survive and carry on, procreate. And so that whole idea is called the survival of the fittest. And actually, there was another guy that no one ever talks about called Alfred Wallace, who also proposed the same theory kind of at the same time, without any collaboration with Darwin. But Darwin is the one that gets all the credit. But there was actually another guy that came up with the same idea. So the father, kind of what we consider the father of genetics, is Mendel, and we'll be talking a lot about Mendel. But it didn't take long, really, if you look at it from a timeline, for us to figure out what DNA was, what our genomic material was, what that whole thing is all about, and where we are today from the time that we've even discovered DNA. It's just amazing what we've... Okay, so let's talk a little bit about Gregor Mendel and his work on genetics and understanding how traits are transmitted from a parent organism to its offspring. So he worked with pea plants. This was a good model for him because there were a lot of traits that were associated with pea plants that he could study, like the color of the flowers, whether or not the actual peas were wrinkled or smooth, whether or not they were green or yellow. There was a lot of different traits that he could look at and that were very simple to observe. So down here at the bottom you can see what I'm talking about, the whether or not the the peas were round or wrinkled, whether or not they were yellow or green, the color of the flowers, the actual whether or not the flowers were axial or terminal, whether or not the stems were long or short, those sorts of things. So he was able to show through his research that the traits are passed from the parents to the offspring in very predictable ways in particular ratios. And so he decided that there was something that controlled these traits, and he called them factors. We now know that they're genes, but back when he was doing his research, they didn't know anything about DNA or chromosomes or anything like that. So he kind of came up with this on his own. And he recognized that during formation of sex cells, or during gamete formation, that the members of the gene pair, the factors, actually separate. So he published his work in 1866, but nobody really paid attention to it. And then, of course, as things go, after he died in the early 1900s, his research was rediscovered. And his research formed the foundation for the study of heredity, which we call genetics. So the chromosome theory of inheritance kind of came about from this research. Once we had microscopes and we could actually look at the cells and we could see chromosomes, it didn't take long to figure out that these factors that Mendel talked about are actually present on the chromosomes. We now know that eukaryote species have a very distinct number of chromosomes, and that these chromosomes come in pairs, and we have two, so we have two of each chromosome, and our cells are considered what we call diploid because of that. And those pairs are called homologous chromosomes. We'll be talking a lot more about homologous chromosomes in the next chapter. Now, cells have to divide in order for an organism to grow, and the two types of cell division are mitosis and meiosis, and this is a big topic in chapter 2 as well. So the chromosome theory of inheritance states that these traits that Mendel observed are actually controlled by genes, and that those genes are actually present on chromosomes, and those chromosomes are transmitted to the offspring through the gametes, which are for humans, are the egg and the sperm. So we're just going to talk a little bit about mitosis and meiosis. Like I said, this is going to be a big part of the next chapter, but it does just kind of give you an overview of this in the first chapter. So mitosis is cell division of just our somatic cells, so our skin cells and our liver cells and our kidney cells, and just any diploid cells in the body. And what happens is you just end up with identical copies of the parent cell. So the parent cell divides into two cells, and those two cells are identical to that parent cell and to each other. During meiosis, that's when you have gamete formation. So this is when we actually make the egg or the sperm cells. And of course, you can't have diploid sex cells, because if two diploid sex cells were to combine to form a zygote, you would end up with twice the amount of DNA that you should have in a diploid cell, and it would be, I guess, tetraploid. So instead, during the formation of gametes, the amount of DNA present is cut in half, and so these cells only have one copy of each chromosome instead of two, and are considered haploid cells. So in the process, you end up with non-identical daughter cells, because they're going to have different copies of the chromosome, and some other shuffling occurs during the process that leads to the content of the DNA. You have the same number of genes, they have the same genes, but they might be different. For example, like the gene for eye color, one egg cell might have blue and another one might have brown, just depending on what alleles the parents had. So this is just a little picture showing what mitosis looks like, and so this is just where when the cell divides, you end up with two daughter cells, both with the same amount of DNA as the parent cell and identical to one another. And with meiosis, you can see it's two processes of cell division, and in the end, you've cut the amount of DNA present in the cells by half, and these cells are considered haploid. So when we talk about, I said that when in the process of meiosis, you end up with these cells that aren't necessarily identical to each other because we do have genetic variation. You can have, thanks to DNA mutations that just occur spontaneously and randomly throughout time, they lead to changes in the DNA sequence that lead to the formation of what we call alleles. And so these are variants of the same gene. So this is a picture showing fruit flies, and one fruit fly has a nasty red eye and the other fruit fly has a nasty white eye, but you can tell that they're different. So this is a gene that codes for a pigment of the eye, and one fly has an allele for red eyes and the other has an allele for white eyes. So it's both a gene that codes for eye color, but these are different variants of that gene or alleles. So as you can see, different alleles are going to produce different observable traits, and these observable traits are called phenotypes. So the phenotype for the fruit fly on the bottom is a red eye, and the phenotype for the fruit fly on the top is a white eye. The actual genes, the actual DNA that's present that codes for these specific alleles is called the genotype, and we'll talk a lot about alleles, phenotypes, and genotypes in Chapter 3 when we talk about Mendelian genetics. So we can actually take mutations and do breeding studies basically with fruit flies or with pea plants or whatever, and actually map out locations of genes based on what the progeny look like by observing these phenotypes in the resulting offsprings. And I think we'll talk about that in Chapter 5 maybe.